All right, welcome to another week of Soul Seminar Online. My name is Chelsea, and today I'm going to be introducing Jaime Simbaqueba, who is currently a science researcher in Colombia. He graduated with his PhD in plant science in 2017 from Australian National University, Yunnan, at Canberra. And during his PhD, Jaime investigated the interaction between soil-borne pathogens, Fusarium, oxysporum, and the solanaceous host plants, utilizing four genetics, genomics, bioinformatics, and molecular biology approaches. In 2018, Jaime returned to Colombia as a postdoc scientist at Agrosalvia. So this is the Colombia Corporation of Agricultural Research. He continues his research career identifying and characterizing new pathogens for the strains in F. oxysporum, in tomatoes and grape gooseberries, and establishing genomics and phylogenetics and phenotypics set tools to study the soil-borne pathogen in solanaceous crops and agrosalvia. In 2020, Jaime joined the Genetic Research Lab in Saichen. So this is the Amazonic Institute of Scientific Research in Colombia as a contractor to set up genomic sequencing of plant species using the Oxford nanopore technology. And with that, Jaime, you can share your screen and get started.
All right, thank you. Right, so are we fine?
Yep.
Let's start. Right, thank you very much for that introduction, Chelsea. So just want to start about introducing a little bit of Agrosalvia, which is the Corporation of Agricultural Research in Colombia, which is in charge of more about the world investigation, about the agro and some transfer research to the agricultural field. So yeah, that's pretty much what Agrosalvia does. So the title of my talk is the genomic resources of Fusarium oxysporum, forma specialis physali, which is a pathogen of Cape gooseberry plants. So Cape gooseberry is the scientific name is Physalis peruviana. It's a solanaceous plant, and it's normally found in the Andean region of Colombia, Ecuador, and Peru, as you can see here in this map. This is mainly the center of, not the origin, but where the plant is normally found. But I also found it in northern Colombia, and also I remember I saw some plants back in Australia, in Western Australia, in Perth. So this plant is important, is cultivated, and you can find it this fruit is important, and you can find it in the supermarkets as a dry product. This is from Australia, and also as a fresh product in the supermarket. So you can just go and find some of these fruit. It is known as Andean superfruit because there's a lot of healthy benefits for the people. So now let's talk about Fusarium oxysporum. This is a fungal species and grouped in a species complex known as Forma specialis, and this fungus has a capacity to infect several different host plant species. And here is an example of different strains that infect, for example, melon, tomatoes, Cape gooseberries, as we saw before, and these banana ones. So they are pretty much related or not, as you can see here in this sort of phylogenetic tree. But what I wanted to mention is that from Fusarium oxysporum, Forma specialis, Lycopersicii, which is known as Fol, we know that the genome contains core and lineage-specific adaptive regions. This also contains genes involved in pathogenicity. And also, these genes are involved in host specificity, and these are located in these lineage-specific regions in the genome. These genes encode for small secreted proteins. In Fusarium oxysporum, these are known as 6 proteins because they are expressed secreted in the xylem, and these are so-called effector genes or proteins. In the tomato and fall battle system, there has been produced resistant cultivars based on plant immunity genes that are capable to recognize some of these 6 genes that are produced by the fungus. This slide I would like to just present. This is from some colleagues in Netherlands from the Martijn Reps lab, and really outstanding work. So they just show that there are some related mobile pathogenicity chromosomes, which determine the host range in the cucurbit host. So, for example, you have here in this graph one strain that infects melon plants, and then the authors co-cultivate with some this strain with some known pathogenic strain, and this strain is capable to transfer some chromosomes that turn this strain into a pathogenic one towards melon plants. In the example B, there is another strain that infects melon plants, which also has the capacity to transfer in cultivation to a non-pathogenic one, and also turn this non-pathogenic into a pathogenic one. In the example C, there is a different strain that infects cucumber plants, and then only one chromosome has been was transferred to this non-pathogenic strain, and turned into a pathogenic strain to several different hosts in the clade of cucurbit. So here, this strain that was not pathogenic, only by getting this one chromosome, has the capacity to infect cucumber, melon, but also watermelon plants, which is the evidence of how or one evidence of how the emergence of new pathogenic strains for plant host species. But let's go back to the vascular wilt of this Forum specialis physali that caused this disease in Cape gooseberry plants. So for Colombia, as I mentioned before, Cape gooseberry is an important plant. There is an industry around that. So you can see here the crops just growing these beautiful capes in the plant, very green, very healthy. As I said, there is an industry. People just depend on this crop. But on the other hand, there is a losses of about 90% in production. And this is the panorama, and you can compare here in this set of photographs. Wilting plants here, you can see in production, but also, sorry, in your right hand, you can see a closer image and a horizontal cortex showing a typical vascular browning of the infection of this kind of fungal species. So in 2010, I remember this fungus was isolated from an infected plant in the field, and some colleagues in Nagrosavia at that time were capable to retrieve the symptoms in glasshouse conditions, showing that this was this Fusarium oxysporum was the actual causal agent of this disease that they saw in the field. And then the strategy to investigate this pathosystem was established around 2010 in Agrosavia. And my group at that time, we collected a lot of plants and generate a genome path of these Cape gooseberry plants. Some of them were tested as a resistant plants, and some of them were tested as a susceptible plants towards this pathogen. So then an RNA-seq experiment was performed with this pathogen, comparing resistant plants and susceptible plants. So early this year, a colleague of mine, Gina Garzon, she published this complete analysis with the set of resistant plants, um, and she found sort of 8 differential expressed genes during this infection that could be related to the immunity in the Cape gooseberry plants. However, on the other hand, I took this RNA-seq data, uh, interaction with the susceptible plants, and then took with me to perform my PhD at the ANU in Australia. So I wanted to, at that time, I wanted to study the mechanism by the Foph that used Foph to infect Cape gooseberry plants. That's the whole idea of my PhD. So what we found there was that in this RNA-seq data, we did some comparison analysis, and with the reference genome of Fusarium oxysporum, which is the full one, and specifically with the chromosome 14, which is the chromosome that is known to be involved in pathogenicity, and also harbors most of the effector genes that have been characterized in the tomato and Fusarium interaction. So by doing this comparison with this RNA-seq data and also with the genome sequence, we found some effectors that were homologous between these two forms specialis. Some of them highly identical, which is the case of these 6, 7, 10, and 12. And some of them not as identical, but for example, here we have the 61A and 61B, which was duplicated in the Foph genome as compared with the Fol one. We tested this process by PCR back and forth. We sent primers to Colombia, and then we confirmed that they were present in the actual strain. And then the next question was to test or investigate the function of some of these effector homolog genes in Foph. So I characterized the 61A and B in Foph. So just to mention a little bit here of the story, this effector gene is highly conserved in several different forms specialis. So here we have 61 conserved in brassicas, a whole set of solanaceous 61s, ones that affect forms specialis that affect bananas, and here we have the ones that infect cucurbits. So here you can see in this phylogenetic tree that we have the Foph 61A here grouped together with another strain that affect Cape gooseberry plants. And here we have the 61B, which is also which is more related to the actual 61 of Fol. So just to mention a couple of details here of 61, this one is involved in pathogenicity, and also 61 knockouts have been produced, or strains and knockout strains with the 61 knockout have been produced. And also that tomato I3 gene recognizes the 61 and confers resistance to the Fol race 3. So we wanted to do a little bit of functional genetics with these homologs 61A and B, and test if either A or B can complement the function of this 61A of the 61 in Fol by transforming a knockout strain with A or B in each of two cases. And also wanted to see if any of these 61A and B could be recognized by the I3 in tomato plants. So a little bit of results here. We have two sets of plants or two different experiments. One using susceptible plants. These are tomato plants, and resistant plants which harbor this I3 gene. So let me explain a little bit here. We have mock plants and plants inoculated with the full wild type, and compare with the full knockout strain. So you can see here the difference when this gene is absent in this strain. When we complemented the strain with the 61A and the 61B, we didn't see this restoration of the virulence in full. So that means that it is possible that 61A and B have a different function, or these are pretty much specific to the Foph strain. However, when we tested resistant plants with this I3 gene, we saw that, typical result here, when we inoculated these plants with the full wild type, there is no symptoms. And in turn, when we inoculated with the knockout strain, we see that there's no interaction between I3 and the 61. So this is disrupted, and then the symptoms will appear again. When we use the 61A complemented strain, we saw that there was no recognition of by this I3 gene. But surprisingly for us, we saw that the 61B from Foph could be recognized by the I3 gene, as these plants are healthy, meaning that this gene could be recognized. This effector gene, this effector gene, yeah. So the resistance phenotype is restored in this interaction with the 61B and the I3 plant, meaning that the I3 tomato could recognize the 61B in Foph from Foph. So that means that there is possible there is a possibility that in Cape gooseberry plants there is an I3 homologous gene that could be interact with 61B. So then I moved to Colombia to continue my work in Fusarium oxysporum, and then I got this collaborative grant late 2019 with Los Andes University, and the whole idea was to investigate the Foph genome sequence that have been produced by that time, and corroborate the findings that I got during my PhD. But also I wanted to set up the functional analysis of the 61A and B in the actual Foph strain that we didn't have the possibility to do it back in Australia. And also do a little bit of effectoromics work to identify or to test if some are genes that have been previously identified could interact with some of the effectors that we have now identified in Foph, to see if we can use this to implement in some breeding strategies of resistant plant of Cape gooseberry resistant plants. However, this was in the middle of this was by the end of 2019, and then early 2020 the COVID just hit the whole world and just stopped the whole research. And also, I just finished my PhD in 2019. Sorry, I finished my postdoc contract 2019. So just the project just really shortened. However, I would like to show you some of my investigation of genomics that I published early this year with this genome sequence of Foph. So here we have the genome assembly. Sorry, I have to mention before that this genome was sequenced by the Illumina technology. So we found 44.7 megabases and 1.8 K context. So we did the whole gene prediction and annotation. And with this sort of data set, we perform my phylogenomic analysis and just corroborate that actually Foph is highly related, well, is grouped in some strain that have the capacity to infect solanaceous plant host. We did some genomic analysis deeper a little bit, and found that the actual Foph strain has the whole core genome regions conserved, but if you can compare with the actual reference genome, there's missing parts of that correspond that actually the lineage-specific regions of Foph. So that means that yeah, it is highly conserved. However, we have this highly fragmented genome. That is because of the Illumina technology that we use. Also, over here, sorry, we have some specific regions that are only present in Foph. In Foph. We also identified putative novel effectors in this genome assembly. So we took these transcripts and we performed research with the Signal P software, and we found 1,400 around proteins, which means that this is the secretome of Foph, or the proteins that have the capacity to be secreted possibly during infection. And 276 of them are effector proteins because these were predicted by this Effector P software. And from this list of 276 effector proteins, we found that 7 of them were novel effectors, which means that these are not present in any other Fusarium strain. And the rest of them were homologous, and we have here, for example, the 6 ones that have been that we identified before. So we also use this RNA-seq implantat that I mentioned before, and we found that 6 of them were expressed during infection, and 1 of them were not expressed at 4 days post-inoculation. And the idea is just then to see one of them could be characterized in future studies. We also perform a manual inspection of the full lineage-specific regions, and we found that actually the gene content there suggests horizontal gene transfer of some of them. So here we have an example of the full chromosome 14, as I mentioned before, which shows the 610, 12, and 7, and up here to this transcription factor alpha, which is highly conserved in these 2 forms specialis. As I mentioned before, we have here 2 different contexts out of this genome assembly, and by manually inspecting, we found the actual that this context is part of this other one, and these also share this cluster of effector genes up to this actual transcription factor. And well, recent evidence now show that in Fol, there apparently these cluster of genes have no effect in pathogenicity, at least for tomato. But interestingly here is that this high conservation between these 2 forms specialis, even the transposon elements that have been actually conserved, we hypothesize that yeah, it's possible that in Fol there is no function, but why these 2 forms studies have this kind of similarity with in the actual pathogenicity region? So that that could be that could mean that in Foph, this could have a role in pathogenicity. We don't know. Or on the opposite, we don't know if Foph is capable to infect tomato plants, or Fol is in turn capable to infect Cape gooseberry plants, which means that this could be well, I mean, this could be the a reason that this set of genes are present in this are conserved in these two different fungus or Furans specialis. Anyway, so next we just check well, or map this RNA-seq data with the genome assembly, and found that a plethora of effectors that were expressed during infection. We saw here, well, the homologs and the novel ones, and the housekeeping genes. And when you compare the housekeeping expression at 4 days post-inoculation with the novel ones, actually 4 out of them are highly expressed at this time point. We also show or found expression of all of the homologs that we found. And interestingly here, the expression of this AV1, that this effector is important, or is used, or is found in another pathogen, and soluble pathogen, which is the Verticillium dahliae, which is pathogen also of tomato plant. And this effector is recognized by another receptor gene in the plant, and lead to the resistant phenotype in the verticillium and tomato interaction. In contrast with the tomato and Fol interaction, this effector is present but is not expressed during infection. So we are now really interested to see if, given that this is present also, this AV1 is also present in Fol, but also we have evidence of expression during infection. We wanted to see if this effector is also or is involved in both pathogenicity towards Cape gooseberry, or there is a new evidence that this AV1 could manipulate the microbiome of the rhizosphere. Well, yeah, getting some advantage of in the case of verticillium, but we don't know what happened in Foph. What if this AV1 has the same effect confers the same effect for Foph in the Cape gooseberry interaction? We don't know. We also just check in each of the effectors that we found, and we saw that these are highly identical with the corresponding part of Fol, but also at the time when we get these results, we got the results of the sequencing of 4 additional Foph strains, and 3 of them were pathogenic, whereas one of them, this number 36, is known as non-pathogenic one. And if you can see here, this table, you can see that this 36, this doesn't have any of the effectors that we already identified for this Furans specialis, meaning that well, there is an extra evidence of the role in virulence, and also that they are only they are present only in pathogenic strains of Fol. So let's move on. So also we found, we hypothesized independent acquisition of the effector genes and the core genes in these Furans specialis. We have here a phylogenetic tree made of 34 different isolates. Among the sequence ones, for example, we have, or in green, I highlight the strains that were sequenced, and we found that actually this this tree shows the two different clades of both strains, clade 1 and clade 2, which reflects the polyphyletic origin of the Fusarium oxysporum strains, which is a typical feature of this fungal species. And in contrast with the effector genes, we have here the genome sequence of the 5 sequence strain, and we see that the effector genes are only present in the in the pathogenic strains, which means that the mechanism of acquisition of these effector genes is different from the emergence of new strains of Fusarium oxysporum. We don't know if, of course, if these effectors are involved in pathogenicity. So this is the whole evidence that we collected out of this genomic analysis or of these new genomic resources of Foph that we generated here. So the summary of the findings, we generated and published the genome sequence of 5 different strains of Foph. We saw that Foph could be grouped in the solanaceous infected strains, which is really interesting to see how this group of strains could be related to each other, share genes or transfer genes each other, and could be a mechanism of the emergence of new virulent strains. And we'll see. And some perspectives is the establishment of the new transformation conditions for the 6.1 A and B to obtain knockout strains. But because of the pandemic, we wouldn't be able to do it. Investigate the role of the AB1 in Foph. We don't know if this is capable to modulate the microbiome. That would be interesting hypothesis to test. Do a little bit of PacBio sequencing to complete the genome assemblies of Foph to get into this chromosome level assembly. And test the possibility of the recognition for the effectors by resistant genes in the plant. So in order to implement to be implemented in breeding strategies for Cape gooseberry, but we don't have sources so far. So I don't know if Rosaria we will continue in this way to research Cape gooseberries. And so last slide. Oh yeah. Last slide, just to acknowledge my supervisor at the Australian National University, the Professor David Jones, at the Resistance Lab, the Professor Alejandro Quintero, which is a co-author of this paper, who is now at the Universidad Nacional de Colombia, and Dr. Carolina Gonzalez from Agrosavia, who is now in charge of the whole Foph and Cape gooseberry research platform there in the corporation. So this is my email, just if you want to contact me, and my Twitter account, just to get in contact. So that's pretty much what I have to say. Thank you very much.
Thank you, Jaime. This is super great. There's a little echo, sorry, let me turn down my volume. So I think like your research is super cool because it spans so much and it's so detailed. So it's really fun to watch and listen to and learn more about what you've been working on. So we are now open for questions. So you can either type them in or just make a note that you have a question. I guess I'll start off. I don't work with pathogens, but I think it's kind of cool what you can do because you can manipulate it and like do these pretty awesome knockout experiments and actually see how it's affecting and things like that. But I feel like I kind of got fixated on like why the pathogen for the Physalis is going to be closer to the Brassicaceae versus the solanaceous is going to be a little bit different. And is that pretty normal in pathogens to have like in that group within plant families that it just like, I don't know. Do you get what I'm saying? So is it pretty typical for strains not to be isolated in plant families or like closely related?
Yeah, that's an interesting question. And I think it's the one of the characteristics of Physalis sporum is it has a broad range of plant hosts. Even though there's strains that infect humans, for example, but these are very different from the ones that infect plants. The thing is that well, given the closer relationship between strains, that means that the emergence of new pathotypes or pathogenicity strains is it's there. We don't know when it happens, but it happens. So yeah, that's that. I don't know if that answered the question. I mean,
No, yeah, I think it's interesting. Do you think it's mostly because of like horizontal transfer happens quite a bit, or do you think it's just like the spontaneous mutations that happen, and that's how it get these
yeah,
different characteristics?
in oxysporum, the main driven mechanism of the emergence of differentiation of new lineages is the horizontal gene transfer. It's the way that you know transfer pieces of sorry, pieces of chromosomes, genes, parts of different genomes. So yeah, this happens. We don't know about the brassicase. I know that the brassicase can restore the 6-1 from brassicase can restore the function of the 6-1 in tomato in the strain that infects tomato.
Okay.
So yeah, that kind of interaction we are talking about. So
do you find a lot of regional differences then? Have you like, is that something, do you find a lot of regional differences? Like
regional,
yeah. So like, would you find the same pathogen, the same genetic makeup? And like, if people are growing the Physalis peruviana in the United States versus in Colombia, what do you think it would be quite a bit different? Or do you think they're pretty stable regionally?
well, this is the first evidence
Okay.
at the genomic level. We sequence this MAP5 Foph strain, and we assemble, and then we start to explore the genome, and then compare with the reference that have been available. And by the end of the project, actually, we've got before we sent to sequence these additional strains, but we didn't expect to have the results so early. And we've got the results actually when the paper was in revision. So we said, no, we don't have time.
Yeah.
We are in the middle of pandemic.
Yep.
We would like to do more, but we just kind of explore the things that we have in the extra genomes.
Yeah.
But then this is an actual field of research of continue investigating.
Yeah.
We don't know. I mean, we don't know about the non-pathogenic one. How the genome is organized.
Do you think the non-pathogenic one could be beneficial? Or is it just like a non-functional pathogen?
Could be, but this is a long discussion. I had this same discussion with some different people, expert people with Fusarium sporum in banana one, and for example, in banana, the risk that poses to use Fusarium sporum as a beneficial microorganism could be a receptor of effector genes and could transform into a pathogenic one. We don't know. We saw the example in the Corbett one, the Martijn Krebs group in Netherlands. Well, they do an amazing job in this, and have really strong evidence about it. So yeah, I wouldn't recommend to use oxysporum as a beneficial microorganism. Yeah.
Yeah, it's exciting. So much. There's always so much more to learn. Okay, so Stacy has a question. I'm going to read it out. So she might have missed it in the beginning, but how widespread are infections by this pathogen across the goldenberry populations? And is there a season where infection is worse, or is it pretty sporadic?
Well, we don't have seasons here in Colombia, so the crop is the whole year you have production and planting anytime. So the main driven of dispersion is the main is the main, yeah. Just infected tools.
Is
When
when?
you use the tools,
Isn't
sorry, is
sorry,
main is a
rain?
labor.
Okay. Oh,
Right.
rainy season.
Like
Okay. Yeah.
the
Right.
labor.
Sorry. Yeah.
So you use the, for example, you walk into the field with your shoes, and then there are spores of fall, and you don't know, and then you just go to the next field or crop, and you just accidentally um, take the spore with you. Same with tools. Same with animals, for example, that live in the area. Can There's no evidence, but there is a hypothetical scenario that possible it is possible that the animals can bring the spores with them. We don't know. This is pretty serious in the banana crops, with this TR4. But same for Physalis. Same mechanism of dispersion, infected plants, infected seeds. Sometimes the fungus can have the can just get into the seeds and then just go around and then to disperse the disease. So yeah, there's a lot of things there. For example, the one tool that we can use in the diagnostics is the new effector genes that are only present in Foph. So using that as a molecular markers, we can just detect spores of Foph in the field. So we can just say tell to the producer, hey, just don't sow there, don't use this plant because it is infected by this fungus, and then just move around. Yeah, that kind of thing.
Can you still sell the crop, or is it like done for once the pathogen starts to like
Sorry,
take over a field? Can you still sell the crop, or is it like done for because you don't want to transport the pathogen?
no, I think with this disease, pretty much done. You just
Yeah.
lose everything. Like just your plants are wilted, so you can do much. So just to destroy the crop and then just move to another land, and this pretty serious practice. Yep.
Do you think you'll expand to just like wild Physalis or solanaceous crop or species to figure out like, does this happen in the wild, or is it mostly like isolated and crop species?
Not really. I mean, actually, Physalis peruviana is kind of wild species. It's pretty recently, like I I can't say if this is domesticated yet. Um, there's no certain about this diploidy. Some colleagues say that these are tetraploid, but there's no evidence. So, well, I don't know. Well, I think the answer has Joyce Van Egg from the Cold Spring Harbor. She's really amazing with Cape gooseberries, so she knows the answer of that. But yeah, actually in Colombia, that where we have, I guess the biggest germplasm of Cape gooseberry, we don't have the answer out of that. And and yeah, there's resistance. As you can see, a colleague of mine explored the resistance Cape gooseberries towards Cape gooseberries, and there is the source of resistance. So we have just to keep on digging and investigating that part. So yeah, we'll see what happens.
Federico, you have a question? You want to unmute yourself?
Yes. Hi. I have a question, and I'm sorry if you already answered. I missed some parts of the presentation, but I've heard that in Agrosavia there is a collection of Physalis peruviana that is diverse. Is it the case? And second, how diverse is it in terms of the response to this pathogen? And are you planning to explore that diversity?
Yes, there's a huge diversity. Well, sorry, I answered the first question. Yes, the Agrosavia has. I don't have numbers here, but I remember there is around 774 different accessions of Cape gooseberries. I think there's more than one species. Physalis peruviana, Solanum. I can't remember the other species. Angulata, and well, yeah, there's a couple of four different species in this year class. My colleagues, I know that they have been tested, lots of them, for sources of resistance towards this pathogen. And there is a different degrees of I say resistance. They only talk about, or normally talk about, tolerance to this pathogen, because they always see this degree of symptoms. Some of them are to me is more resistant, plants more resistant to this pathogen. They say tolerant. But I mean, these degrees of range of symptoms, some of them are more like you see very clear, defoliation, wilting as well, and some of them are pretty strong. They stay there. They just manage to support the infection. I don't know to what extent they have classified there, but yes, these are on a scale of resistance, to more resistance to less resistant, and scale of different strains of Foph, from hypovirulent, non-virulent, hypovirulent to hypervirulent. So, for example, the one that we sequence, that we explored, is one of the hypervirulent ones, the most virulent ones. So yeah, Agrosavia has the whole resources, I mean natural resources, to investigate this, to keep on investigating the pathosystem. So I don't know if that answered your question.
Yes. Thank you very much.
That's right.
So Eva, do you want to unmute yourself and ask your question?
Yeah, yes. Hi, thanks for the nice talk. I have maybe I missed it, but so you mentioned in the beginning your transcriptome of the Fusarium, you took it from infected plants, right?
Yes.
Or so you also have the plant genes, or what did you do with these genes?
Yes. I didn't do anything with the plant genes. I just took away from the whole RNA-seq, and then took the reads that belong to the actual Fusarium species.
Okay, but you have the data.
Yes.
Of the plant genes as well.
Yeah, I forgot to mention that the whole data, the RNA-seq, is now on the gene bank. I don't know the actual accession, but I remember Gina Garzon from Agrosavia. She told me once that they put the RNA-seq in the gene bank for researchers to explore.
Okay, I will look for it. Otherwise, can I ask you if I can't find it?
Yes, exactly. Yeah.
Okay,
Cool.
good. Thank you.
That's all right.
Does anybody else have a question? Raise hand. All right. Well, if that's all the questions, thanks again for presenting. Next week, Rocio, do you have it up for next week? Let's see who gets it faster. So next week, of course, we have another presenter. Let me see who it is. And if you still have lingering questions, Jaime is definitely available via email. As he said, he gave you his email and Twitter, so feel free to contact him. Next week, we have Marco Cuba Menchigo. He's talking about the advances in taxonomy and phylogeny of Lithianthias from Peru, so that should be pretty great. So definitely tune in next week, and thank you guys for all attending, and have a good week, good weekend.
Thank you very much.
Thank you, Jaime, Chelsea. It's great.